the key people of this study, Professor Paola Brun, me, and Professor Roberto Vettor. And in this slide, other collaborators for the future development of the study. Antonella Pasquato, that is here in my room, Laura Cendron, and Eniza Muner. The idea of this study came from my daughter, Marta, that received the diagnosis of KAT6A syndrome 4 years ago. In the genetic evaluation of Professor Romano Tenconi, there was this sentence: the mutation in exon 15 results in a signal that interrupts the synthesis of KAT6A protein, which is shorter and consequently is discouraged. Considering that Marta possesses one allele encoding for functional KAT6A, but the other allele being defective, I thought that the biological mechanism and all the substances able to upregulate KAT6A should be investigated. And the question is, can we stimulate KAT6A expression? During literature search, I found this paper published in Environmental Pollution Journal, in which bisphenol A was indicated as a component able to upregulate KAT6A. But bisphenol A is contained into plastics and is a toxic substance. So I looked for some other compounds similar in their formula to bisphenol A, but without any toxicity. And in this panel, you can see all of the molecules that were used in the study. There are different hormones: progesterone, or testosterone, hydroxyprogesterone, one amino acid, tyrosine, and two molecules, luteolin and erydiol. And only these two last reported molecules were purchased, because this research started without funding. So this is the scheme of our experiment. Luteolin or other substances were used to pretreat human cells, in particular primary human osteoblasts. And then the total RNA was extracted, and KAT6A gene expression was quantified by PCR. And as you can see, there is an upregulation of KAT6A induced by bisphenol A versus the control. But there are two not substances, hydroxyprogesterone and luteolin, that produce an increase in KAT6A expression by 4 and 2.5 folds respectively. So these results were published in the Journal of Rare Diseases. It is an open access journal, so you can see the results online. But to go further, we go back to the initial question: can we stimulate the KAT6A expression? And does the modulation of this gene expression represent a good approach for the delay of the symptoms and for the reduction of the severity of the disease? And it is a good point to see that my work started in 2020, but in December 2022, a group of Spanish researchers published another paper in which pantothenate and carnitine were found to reduce pathological alterations effect in cellular model of KAT6A syndrome. In particular, with the pretreatment of pantothenate, there is an increased production of acetyl coenzyme A that, together with KAT6A, produce the acetylation of histone. This treatment with pantothenate is able to improve gene expression of KAT6A, but also of protein expression. And more important thing is that this picture improve the normalization of histone acetylation. The acetylation of histone is very important for chromatin folding. So if the question is, does modulation of KAT6A gene expression represent a good approach for the reduction of the severity of the disease? The answer may be yes. We can induce a modulation, upregulation of KAT6A, both using its coenzyme A, or as we have done, using luteolin to improve the upregulation of KAT6A. So our idea is to produce more results to confirm the preliminary results reported in our paper. So our short-term goal is to complete the research testing KAT6A gene expression induced by luteolin, not only in human osteoblasts, but in different human cells. And the second point, to check the expression of KAT6A protein after luteolin pretreatment, both in healthy and in patient cells, in order to compare the behavior of these cells. And third point, to carry on functional studies for example, to test histone acetylation. The budget for this part of the study is about $25,000, and we are looking for funding in order to perform these experiments. But we have other ideas for longer-term goals, and for this two slide, I give the stage, give the floor to Antonella Pasquato.
Okay, hi. Yes, I joined this project later on, mainly because my background is really on enzymes and high-throughput screening. I found this topic extremely interesting. When I started to look at the background, what this KAT6A is, we found essentially nothing really much, very few information. Most of the information are linked to tumor. Essentially, KAT6A seems to be upregulated or dysfunctional somehow in tumors. But what about this rare disease? How does it work? So this is the first question that we would like to answer to. And to really answer properly to the question, we really have to dissect the protein. We know already that the protein has a binding motif to DNA, another one that is more catalytic, and the cytosolic tail, the terminal part, that is really the unknown part. No one knows what it does exactly. Very importantly, something that I found just looking at the literature, most of the patients that are affected by this syndrome, instead of having pine mutations, they have truncation. Also, Martha, for example, she has a truncation, and the entire catalytic domain is preserved. So theoretically, it should still work, but actually, it does not. Why? How it does work? Again, there are really confusing or really not appropriate information. For example, some papers report that patients carrying mutations on the KAT6A, they don't express any protein. They are not able to find the protein itself. Another paper, the one that Monica just mentioned, the Spanish groups, they show that the protein is there. So who is right here? How does it work? I personally believe that each mutation, each truncation has its own way of being in cells. So just to know exactly how the protein does look like, this is extremely important because it gives really key information to know how to proceed. Now that there are all these techniques, mRNA, all these new screenings and everything, imagine what we could do if we knew exactly how the protein does look like, how it works. So this is really the very first key information that we want to dig out. Then the second part is on the activity of, let's say, on the enhancement, improvement of the basic normal level of KAT6A activity. So this is what Monica has done. She's really very good. She got some information. We are starting thinking, this is why we are collaborating also with the biochemistry, biology department. We believe that this signaling upregulation via luteolin may be these are all hypotheses to be tested. Estrogen receptor could be involved. So again, this is an entire pathway to really dissect. There is an entire system to really study in depth to know how this works. And this is another big part of something that we would like to do. And finally, we would like to have information on exactly how this KAT6A does work. We know that it should activate transcription factors or other genes. Which are these genes that are regulated by KAT6A? If we really, let's say, if this is the normal level of KAT6A, if we exceed a little bit, do we have side effects? Which type? What we have to look at? So this is the entire an entire part of the project, and this is really to set the ground, to know the basic information on the system. And we are looking for funds. This is really long terms, and we are really very aware that it's not something that can be done in one day. Another part that we would like to do is something unrelated. It's more applied research or translational research. Here it's really screening. It's where I have more, let's say, background. I'm an enzymologist. I'm studying high-throughput screening. When I saw this enzyme, I said, okay, let's see, we can do something here. So the first thing that we can do is double-blind screening. So we don't know, it's a little bit what Monica has done, because we don't know the receptors or anything on the pathway. We have no idea, and we screen libraries of compounds. What we do is simply to test for expression of KAT6A. So it's very simple. We have to start the experiment, of course, but this is really copy and paste from other known systems, so nothing really much to think about. There is a luciferase reporter. Whenever you have something that trigger the KAT6A gene expression, you have a positive signal, so you can read the luciferase. Again, you can, if this estrogen receptor is the good candidate that is really linked to the expression of KAT6A, you can do screening looking for ligands, appropriate ligands for this receptor, looking also at side effects and all these things. So the assay has to be really carved in an appropriate way. All the enzymatic part, this is another screening that can be done. Here we have the entire enzyme can be produced, is soluble, so can be expressed, purified, and then with appropriate substrate you can provide compounds and screen and do whenever you get inhibition of the catalytic activities. And finally, something that is, let's say, complementary to all these that are really hands-on, I mean, wet lab experiments. There is all the in silico part where you do screening simply, maybe with the help of now there is all this artificial intelligence softwares, and you can compare the backbone, the structure of luteolin with a library to see if there is any match and to find something that could somehow work. So these big branches, either fundamental research, basic research to know how the system works, because nothing has been done so far, is extremely important because it can open really the doors to I think that all these techniques are coming, like the RNA and so on, but we have to know what we are dealing with. So we need to know KAT6A a little bit more. And then all the other part that is screening for compounds able to regulate the expression of KAT6A. We are actively now searching, looking into possibility to find funds, to fund money to get this work done. Okay. Do you have any question, anything? No one. Can we?
Okay. Thank you.